You can do, like, if you have a document or if you want to do your whole screen.
Yeah, I don't see it. For some reason, I don't see that PowerPoint as an option.
It's not letting you do PowerPoint. There should be,
Yeah, I don't know if you got that message, but like the usual classic Teams is unsupported, so do you have to like switch to the new one now?
so it's not letting you do it. It's not letting you share.
Yeah, I don't see the PowerPoint as an option to share.
If you want to send it to me real quick, we can try that. Sorry.
I just admitted you from the lobby.
Let's see.
How do you mute your control sound?
How's this? Is this working?
1 second.
It says it's preparing it says it's loading my presentation.
Yeah, I think it is. It's just taking a second here. Yeah, okay. There we are. That's
okay, great.
There.
Okay. Sorry about that.
Sorry. No, you're good.
So thank you for having me to speak. I wanted to address the group because I think we have some updates, and I know that there's probably a lot of new people as well, and just to sort of share what we know, what we don't know, which is a lot, what we're doing, and then to take questions. And I'm going to sort of talk about the first two things, and then I'm going to pass things off to two very talented postdocs in my lab, actually jointly shared by myself and Chris Mackinson, who are really leading the work, and I'm going to let them talk to you about what we're doing. So I'm a medical doctor. I see patients with pediatric patients with epilepsy, but I also have a laboratory and I'm trying to figure out how genetic disorders cause epilepsy and how we can do better to treat patients. And so one question that comes up a lot that I always like to cover at the beginning, especially if there are families that have just recently gotten a diagnosis or have lingering questions about this, is how do we know? Like how you get a genetic test result back? How do you know that that's the answer, that that's what's causing symptoms? And the answer to that is that, our genes and genetic differences are what makes us all different. That's why we all look different and things like that. But there are certain genes that don't change. They don't change between individuals, and they don't change in some cases even between species. They're highly conserved, is what we say, even to other types of animals. And that's because the genes do something so fundamentally important that it's critical that they be exactly like they are, and they really can't change. And CSNK2B is one of those genes. And so the variants that we see in patients, affected individuals, we don't see those in large databases of healthy control populations. And oftentimes, although there are exceptions to this that we're going to talk about, oftentimes it's a new change in the gene. So it's a new problem in the child that wasn't there in the parent, and it goes along with a new change in this gene that really doesn't change from person to person. And it turns out that there are a lot of genes like this. And you might sort of expect that because the brain is really complex and a lot of things have to happen in the right ways. It takes a lot of different genes, making a lot of different gene products to function properly together. And so there are hundreds of genes and hundreds of thousands of variants in those genes that can cause epilepsy, intellectual disability, autism, things like cerebral palsy, and other neurodevelopmental disorders. And this is a schematic showing how this sort of unfolded over time. And this is going to be important. We'll come back to it in a second when we talk about why CSNK2B is the most important epilepsy gene that nobody's heard about. But in the beginning, the genes were sort of identified in families. So inherited genes, inherited variants being mapped in families with very rare conditions. And then you can see that around 2009 or so, something started to change. And that was because next generation sequencing allowed for the identification of these new changes in genes and led to a wave of gene discovery that's been going on since that time. And so why do I say that CSNK2B is the most important epilepsy gene you never heard about? Well, just as a couple of examples. One is that there was a research project here where 737 individuals with epilepsy were sequenced to identify causative genes for their epilepsy. 68 of those 737 patients got a genetic diagnosis on the basis of that sequencing. But there were 48 different genes. So this is exactly what I was talking about before, where there's all kinds of different genes that can cause epilepsy and neurodevelopmental disability. Only 10 of those genes were seen in more than one individual. So there were some more, among the rare, there were some more common among the rare. And, for those people in the field looking at this, people who have been doing research or clinicians who have been treating patients with genetic causes of neurodevelopmental disability, most of these genes are not, they're known, they're well known, they're even sort of becoming known in the lay public to some extent. SCN1A is the cause of Dravet syndrome, which is probably one of the most common, severe epilepsies in the pediatric population. DEPDC5 is a major cause of focal epilepsy that leads to something called focal cortical dysplasias, which are the most common structural cause of epilepsy in kids. KCNQ2 causes the most common form of neonatal onset epileptic encephalopathy. SCN8A is a sodium channel that's also a well-known epilepsy gene. CACNA1A is well known. KCNQ3 goes along with KCNQ2 as being some of the oldest sort of described epilepsy genes, and so on and so on. In the list is CSNK2B, which most people in these circles had not heard about. And I'm going to tell you what we're doing in terms of increasing education in a minute. So it's the one that stands out like a sore thumb in a way. And a study of patients with status epilepticus, which is when patients start seizing and they don't stop and they have to go to the hospital and for major medical management. Looking at the genotypes of those patients, the ones that had genetic causes, there were certain genes that popped out. And again, these are recognized genes. SCN1A again, MECP2 is the cause of Rett syndrome. So a lot of people know what Rett syndrome is. It's a very common neurodevelopmental disorder in children. And there's SCN8A again. And then there's CSNK2B, which again sort of sticks out like a sore thumb in being less well known. And why is that? So the reason that is the case is because, are you seeing my notes too? No, okay, all right, that's just, okay. The reason that this is the case is because the first cases were first reported in the literature as late as 2017. And I told you that the explosion really started around 2009. So 2017 is kind of late. And the first reports, there was 2 patients reported by the French group, which is where the term that's been used to describe the condition Pobens comes from the first and last names of the authors of this initial report. And they described some pretty severely affected individuals with global developmental delay and one with myoclonic epilepsy. And that was very quickly followed also in 2017 by a Japanese case report of a patient also very severely affected, also with myoclonic epilepsy. So it looked like there was a sort of consistent phenotype being reported in 2017, but only a couple of patients. In 2019, 2 more patients were reported with CSNK2B. And for the first time, a different type of variant was reported, which is called a missense variant that we'll talk about in a minute. But up until that point, they were all sort of variants that were expected to affect the expression of one of the copies of CSNK2B. This was different. And then for the first time in 2019, a series of 9 patients were published, which was to that point the largest group of patients published. And so at the start of the pandemic, there was really 14 patients published. And so that's really, that's been the sort of slow trickle until we reported 25 patients from the US. And at that point, it became clear that not all the patients have epilepsy. That was sort of known from the beginning, but in addition, that some patients weren't as severely affected as the initial reports. And even more recently, there is a nice series from an Italian group that I happen to be friendly with, who reported that they agree that there's a lot of variability, which was sort of the thrust of our paper, but they highlight the fact that even within a family, there are cases where different individuals are affected differently with the same variant. And so this becomes very important. The two sort of trends that come out of the literature are, number one, there's something called ascertainment bias, where the first patients reported are the most severe, because those are the ones that the researchers want to sequence, because they know that they're going to find something, or they feel like they have a higher likelihood of finding something. And over time, as more and more patients get sequenced, and especially as it rolls into standard clinical care, then we're sort of discovering milder cases over time. And that's something that's true, I think, across the board for all of these genes. And then there's the phenotypic variability that comes out. And we certainly found that in our group of patients, but the Italian group really hit the nail on the head by showing that even within a family, the same exact variants can cause very different conditions. I think one sibling had ADHD, and that was it. And then the other child is more severely affected. So quite different. We're going to come back to why that might be the case, and I think that's a major question, a major research question. So I don't want CSNK2B to be not known. We want to get the word out about it, and we authored a website for the Child Neurology Foundation. So CSNK2B is represented there. There's information there for families, newly diagnosed patients. And I'm happy to say that after almost a year of working on gene reviews, we have a summary of the patients and what's been reported to date in the literature that should be published soon. We're making the final edits that they requested on that. And that's what I want to share with you next, is what we found by doing that extensive literature review with the gene reviews. So there are now over 80 patients reported, but like a lot of the patients show up in big cohorts and there's not a lot of information. Some of the patients are reported, but they're still 2 years old. So we don't really know the full manifestations, how they're gonna do. But here's what we can say. So most patients with pathogenic variants in CSNK2B have developmental delay. and most of them have both motor and speech affected, and so typically would fall under the rubric of global developmental delay. For those that were delayed in terms of their motor development, the average age of walking was about 2 years. It was pretty infrequent for people to be walking. This is true across the board. When you look at motor development, the longer it takes you to do something, the less likely you are to do it. And so patients typically were walking by 5 years or not walking. Speech delay was really common, but most patients did end up speaking, with a notable sort of exception of over 10% who remain nonverbal or minimally verbal. And so you can see that that's sort of touching on this spectrum. For the ones that were over 5 years of age, that you can kind of assess their cognitive function, most had some level of intellectual disability, but it really ranged from borderline, which many people would say that borderline is not intellectually disabled, it's borderline. And then there were patients who really did not meet criteria for cognitive disability, intellectual disability, but many of them had a learning disability. And so again, quite a spectrum of severity. Most patients had seizures, but some of those seizures were like one-off seizures or febrile seizures. Not everybody had epilepsy, but if you were going to develop epilepsy, the vast majority, 90%, developed it by 3 years of age. So we're looking at really an early onset epilepsy when it does show up. But the type of epilepsy is very variable. Certainly the myoclonic seizure phenotype keeps coming up again and again, like it did in the initial reports. But that's not the whole story. There are lots of different epilepsy types, seizure types, and a lot of variable response to medications, with some patients having to go into the hospital repeatedly for status epilepticus, which is why it showed up in that study. And then there was a smattering of other issues, growth issues, coordination problems, low tone, particular facial characteristics, particular issues with fingers and toes, and then differences with the way that the teeth develop. And again, those were sort of not, they're not across the board, but they happen enough that we know that it's related to the condition. There were other things that show up because we're taking a close look at these patients, but it's not really clear if the rates of those differences are higher than in the general population or not. And so it still remains to be determined whether some of the cardiac things have been reported, for instance, whether those are something that is a real finding, or just because we're looking very hard at patients, we're finding lots of little things. This is just a picture of the epilepsy incidence sort of cumulatively over time to show you that it really starts early, most of the time when it starts, with more than half in the first year, and even more than half in the first 6 months. This was from this is not all epilepsy, but this is the epilepsy in our paper. So we don't know how variants in CSNK2B cause disease, and we don't know why patients are so different from one another. But as a sort of introduction to the talks that are coming, CSNK2B encodes something called CK2 beta. It's the beta subunit of a kinase. So a kinase is a type of protein, and in this case, the kinase is formed of more than one subunit. So each subunit is a separate protein that come together as a complex. And you can see that it's sort of shaped like a butterfly. And the beta subunits, which are encoded by CSNK2B, they make the hind wings of the butterfly. Whereas alpha subunits that are encoded by a different gene make up the forewings of the butterfly. And the beta subunits also hold the whole thing together. And it seems like the CSNK2B is expressed everywhere throughout the body, but it is very high in the brain. And it's a kinase, which means that it phosphorylates other proteins. So that means that it runs around sticking phosphate groups on other proteins. And so if you can imagine, and in this case, it does it to a lot of different proteins. And so when proteins have phosphates on them, phosphate groups, when they're phosphorylated, they behave differently. It's like a different mode. And so it changes kind of the behavior of large groups of proteins, kind of shifting things into a different mode of action. So if the little people in your house at some point during October run around and start putting up skeletons and carve pumpkins and decorate things with spider webs and all of this kind of thing, and you come home and you walk in, you say, oh, well, I see we're in a different mode now. It must be fall, it must be Halloween. We must be doing the Halloween things now. And so that's kind of how I think about how kinases have the ability to sort of shift the mode of how a cell operates. The variants that cause disease in CSNK2B do different things. So on the left side, you see what happens in most people, unaffected people. We have 2 copies of CSNK2B, and it makes a full complement of the CK2 beta protein below. So 2 copies, twice the amount of CK2 beta. If a person has a type of variant where the expression from one of the genes, one of the copies of CSNK2B is affected, so that it's not being made from one of those copies, then you wind up with half as much. And that's a condition called haploinsufficiency. And we know that that's one type of variant that can cause symptoms in CSNK2B. The other type of variant is called a missense variant. And so again, the change to the gene is only in one copy. So that's what the asterisks indicate in the DNA section of the diagram. And instead of affecting the expression so that one copy isn't made, instead what happens is something different is made. So a different version of CK2 beta. And that different version may not work. It may work less well. It may cause problems because it affects the formation of the larger complex, the kinase itself. And so it can be potentially worse than not making enough. Or it may do something completely different, like the beta subunits of the kinase may determine what gets phosphorylated, and the mutation to the protein may make it so that the kinase targets things that it should not target, other proteins. And so one answer to why patients might be different would be the different genetic variants that we're talking about. But that probably is not the full explanation, because I told you already that we have patients who have the same variants who are very different from one another. And we even have patients from the Italian group who are directly related to one another, who are quite different from one another. So it can't be the full explanation. what about the epilepsy? So one possibility for some patients, and it's not true for most patients, but for some patients who are very severely affected, the CSNK2B variant, which is causing some degree on its own, causing some degree of neurodevelopmental disability, is also causing epilepsy. And if the epilepsy is very bad and the seizures don't stop and the electrical activity in the brain is very impacted, that has the potential to also affect development. So I think that this is also a mechanism, but I think it's relatively restricted to patients with very severe seizures that are not stopping. So probably only applies to certain cases. I think, you know, the current thinking right now is that CSNK2B as a disorder is just incredibly variable in how it manifests across different patients. And that likely has to do with the fact that the patients themselves are different from one another. And so there's something happening at the level of all of the other genes that people have that make it so that how it manifests in one person is not the same as how it manifests in another person. So what do we need to do next? So we need better information, more information on patients, because the variability that we're seeing is really a problem. You know, not being able to predict what to expect based on the variant makes things very challenging. We need sort of deep phenotyping data, meaning that we need the quantitative data that is collected on patients in clinical care, like the EEG data and their neuropsychological data. And to that end, we've been in contact with 17 previously unreported patients, and we're working to get EEG on folks. And I mentioned that I know that the Italian group that published is kind of a funny coincidence. There's so many coincidences with our work with CSNK2B, but one of the coincidences is that the person who got me interested in genetic epilepsy from clinically and also research trained with that same Italian group. And so they're basically like cousins. So as soon as I saw the paper, I reached out to them, and they're going to give us their EEGs. So in terms of the laboratory, the bench research, what do we need from the work? So we need model systems because we need to try to understand how the variants cause symptoms. Are the missense variants behaving differently from the ones that affect expression? Why are the patients so different? If there are genes that are making the variants, more severe in certain patients or less severe in certain patients, that's an important clue, perhaps, to how to help patients. What's the relationship to patients with the related condition affecting the alpha subunit of the kinase, which we still don't know. And we need model systems as a way of testing treatments that we develop, new therapies to target the underlying genetic variant. And so I'm going to have two postdocs in the lab talk. First, Iya Pritkova is going to talk about human cellular models. We have 14 patient-derived lines. Each has a unique variant. They're all healthy and growing. 9 of them were collected last year at this meeting. So that's amazing. And it sort of reflects the literature in the sense that about half are going to are expected to affect expression of CSNK2B, and about half are missense variants. So that's wonderful. And we're currently turning those into stem cells to be able to make nerve cells and even mini brains in a dish that's going to allow us to really ask what happens to the cells and nerve networks of actual patients. It's a long, costly, and challenging project, each line has to be corrected to make its own control. And then the next person who's going to talk is Abigail Carbonell. She's working on mouse models of CSNK2B. And with a whole mouse model, we can ask what happens to brain function when there's not enough CSNK2B. And we have a conditional model that allows us to control which cells we impact with the mutation. And we're currently characterizing 4 different approaches with this mouse, 4 different mice essentially. And that allows us to look at behavior and development as well as seizures and EEG. And now I'm gonna stop sharing so that Ia can present. If I can figure out how to stop sharing. Oh, there we go. Yeah, it's all you. You're muted.
All right, hi. Everybody see the screen okay? Yes? Okay. So I am Eber. Thank you, Tristan, for the introduction. As Tristan mentioned, I am leading the side of the project that is concerned with the human patient lines and other ways in which we can use human models to study disease. So to reiterate, we have 14 patient fibroblast lines, and they contain a number of different mutation types. And some of them we can see cluster in the early parts of the gene, whereas others in the later one targeting different functional domains. So we have a pretty good spread of variability in this cohort. So our first order of business was to characterize expression. So what you're looking at here is a plot of RNA. So that is the gene product that gets transcribed across our different patient lines. The full change in expression relative to control. So if it's above 1, it means it's higher. If it's below 1, it means it's lower. So right away, we can see that there is quite a spread in variability between different mutation types. And just because like it's the same mutation type doesn't mean it has the same effect. So we see both loss of function in terms of transcription as well as some putative gain of function variants. And then there are some that so far don't seem to have a difference in expression. So that means the changes are just happening at a different level other than RNA. So we are in the process of replicating this, incorporating our new patient lines to further characterize this cohort at both the RNA as well as the protein level. So to come back to the point of variability, it is a big challenge in science because we like to have our experimental condition, our control condition, and only change one thing, whereas humans just don't work like that, right? We may look similar, but all of us have like different genetic backgrounds and different ways that they interact with the environment. So there are 2 main ways that we can work with that in the lab. So having identified the gene variants, we can first correct the mutation, creating one type of isogenic pair, or we can introduce that same mutation to the other control lines. And that way we can dissect the impact of the variant away from individual backgrounds. So we have designed prime editing approaches to introduce and to correct the variants present in our cohorts. Shout out to B for leading the charge on that. And we have selected 4 variants with the most prominent loss of function as our first cohort for studying of CSNK2B mutations. And we already were able to introduce one of them to a control cell line. So we already have an isogenic pair. And then these 4 have also been submitted to the Columbia Stem Cell Core to induce them into stem cells. In addition to a patient-specific loss of function, we are inducing loss of function in healthy control cell lines using a technique called RNA interference. So we have 4 cell lines derived from unaffected controls, and then we use a small piece of RNA that essentially prevents the translation in the making of the CSNK2B protein, and we introduce that across the board, thus creating another type of isogenic comparison. And with that system, we can induce neurons and study the effects of just general absence of CK2B activity on how neurons grow, their shape, as well as how they function, how they fire simultaneously, and how excitable they are in response to stimulus. So there are several models of neuronal development that we plan on working with. So we have early development here, where we look at the formation of neural rosettes. So that's before, like the earliest stages of brain formation. We also have a middle stage development with an induced excitatory neuron system growing in 2-dimensional culture. And we can use the system to kind of measure a time course of how active the neurons become as they mature. And we use something called a multi-electrode array to keep track of that. And finally, for an outlook on a more mature brain, we can perform a high-resolution single-cell activity assay using 2-photon imaging, where we can look at how mature cells function together in network. So we've already conducted a pilot experiment to that end. We had 4 control cell lines treated with RNA interference, the isogenic pairs, just like I described before. And then we used calcium sensing fluorescent dye. And this is an example field of view from the microscopy experiment. So you can see different like cell bodies here, as well as projections between them. And by identifying like specific neurons of interest, we can then measure how these function over time. So you can see that this is just one recording and several example ROIs from one little organoid. And this one in particular has been treated with CSNK2B knockdown, the interference condition. And we can see that there's quite a bit of spontaneous activity occurring in burst patterns. So the next step for this would be to analyze all of the traces from all of our conditions, and then we can measure things like spike frequency, prevalence of bursts, and then response to electrical stimulus as measure of how neurons communicate with one another. Another exciting direction for this project that is underway is a collaboration with Dr. Daniel Schwartz at the University of Connecticut, where we are going to be looking at the phosphoproteome. So we can actually see which targets are affected by different modes of CK2B. And as Tristan explained in his introduction, protein phosphorylation, it is very important for the function of the cell. So based on what parts of the protein are phosphorylated or not, that determines whether the protein, like where in the cell the protein can go. So that can affect things like neurotransmitter receptors or ion channels that also control neuronal excitability. So by for our first pass at this, we've got our isogenic conditions where we are going to be comparing just variant-specific versus generalized loss of function of CSNK2B. And by doing so, we can identify overlapping and non-overlapping changes in protein phosphorylation. So anything that we see like in the yellow circle, we would know that these phosphorylation targets tend to be CSNK2B independent. And then can identify variant-specific effects and then generalize non-specific effects. And perhaps by focusing on a set of targets that overlap between variant-specific and generalized loss of function, we can come up with a more generalized therapeutic strategy. So to quickly summarize, we are trying to leverage these human-induced pluripotent stem cell models for personalized genomic medicine so we can understand patient-specific neurodevelopmental, cellular, and molecular events and can identify the appropriate course of treatments, a drug selection, and with these assays, we can develop personalized gene therapy in vitro. We of course, have the approach of prime editing mutation correction, but for a series of edits that are all clustered together, a different approach, such as an exon replacement strategy, may be more. And then finally, like the real power of this approach is the isogenic design that allows us to build in like the natural human genetic variability into our model and hopefully make our research translational. So that is it for me. Thank you so much for your attention. I will stop sharing and let B take over the behavioral front.
Thank you, Iya. That was awesome. So with these human cell models, you know, we're going to be able to see how CSNK2B variants change the biochemistry of these nerve cells, as well as the way that they talk to one another electrically. And that's going to give us targets to go after with therapy. And it's going to give us a sense of the answers to some of those questions about how it all works and why there's so much variability. And now Abigail Carbonell is going to talk about the work that is being done in the lab with the mouse model.
Great, thank you so much for the introduction, Tristan. And I think that both you and Dr. Friedkova have given a great introduction to the mouse work. So I'll go right into where we are with that project and the types of results that we've been having. So as was reviewed, we're talking about CSNK2B neurodevelopmental disorder. And here I just want to focus on three different parts of the discussion. One is that the types of, this is a rare genetic disease with neurodevelopmental features, and it was first identified with variants in spliced site mutations. And furthermore, predicted loss of function variants and missense variants has been described or have also been found. The types of genotypes that have been found have been heterozygous variants. Family members that are on this call will know that most of the, if not all, of the variants identified so far have been heterozygous. Only one copy of CSNK2B has been affected. And overall, when we talk about predicted loss of function, in one sense, we can group these all together in terms of loss of the start of the gene, nonsense, which is early termination of the gene, frame shift, which usually does cause early termination also of the gene, as well as differences in splicing, the way the gene is put together. Even missense variants, if they're not predicted to cut off the gene entirely, could also affect the function of the kinase to the point that it is a loss of function of the protein, even if it's not loss of function or loss of expression of the gene. And then the main phenotypes, as has been described already today, have been seizures, differences in learning and development, as well as some features that have been found in some families, like autism, ataxia, which is differences in motor balance and coordination, and hypotonia, which also causes difference in movement that can affect gait, walking, and daily function. So the different mouse models I'll expound on here. In collaboration with the Mackinson Lab, we've developed this CSNK2B floxed mouse. Essentially what this does is tag the CSNK2B gene in the mouse to be targeted or deleted by the CRE enzyme. So that's our foundational mouse model. Then we can cross it to different and existing CRE lines, which express the CRE enzyme in different regions of the body and also different times. So one of our approaches is the SOX2 CRE, which targets the CSNK2B gene throughout the body. We have this on 2 different genetic backgrounds. As this has been mentioned, genetic variability is important, and we want to make sure that what we're finding isn't limited to just one mouse model or one strain, but on different backgrounds as well. The ICV CRE targets CSNK2B in the central nervous system. That's done by injection into the brain. The EMX1 CRE is targeted in excitatory neurons of the forebrain, including the cortex and hippocampus. And the GAD2 CRE model targets CSNK2B inhibitory neurons. These are neurons that slow down activity in the brain, and problems there can cause seizures. It can cause differences in development as well. So this is an overview of the types of testing we can do in these mice. Patients are not mice, but there are different big categories. We can categorize the findings in our patients that we can test in mice. For example, in development, we can look at growth and survival. We can look at developmental milestones. I know that this has been a topic that's familiar to families as development is tracked in kids and families over time. Behavioral tests, we can test activity and exploration, motor coordination, which we already mentioned as can be a difference or a problem with CSNK2B, as well as learning and memory, which can be analogous to intellectual disability and learning disabilities, as well as acoustic startle. Sensory motor reactivity and modulation is what's tested there. And then when it comes to seizure testing, we have a couple different types of testing, including electroconvulsive threshold and the electroencephalogram, which is used clinically in patients to look for abnormal electrical activity, and we can also do that in mice. So the questions that mice can help us answer is, does loss of CSNK2B cause changes related to the clinical features in patients? Which regions and cell types are responsible? And what are the best targets and models for therapy? So I like this slide because it sort of reinforces what Dr. Friedkova already showed in terms of what's found in the patient lines. So just like in about half of the patient lines, we find a reduction in CSNK2B expression at the RNA level. In our mice, when we look at different areas, I've now done this experiment in cortical tissue, which is from the front of the brain. I've done it in body tissue from tail and ear in the mouse, and I've also done it from other brain regions that are not cortex and hippocampus. I've found similar results in our mouse models, which is that there's about 40%, roughly half the amount of expression in these mice compared to controls, which shows that this is a good, at the RNA level at least, these are a good model of many of our patients. I'm going to be showing some graphs, but mostly I'm just going to share a take-home message and just which lines and which points of the graph are going to be important for making the point that we're making for these mouse models. So this is SOX2 CRE. What's targeted here is the whole body, CSNK2B in the whole body of the mouse, heterozygous because only half of the gene expression is affected. And you can see from this red line that there's a significant drop-off in survival over time. So CSNK2B in the whole body is important for survival. Interestingly, we see the same thing when we target CSNK2B in the brain only. In this model, it's brain focused, the ICV CRE, and you can see that the homozygous, which is missing even more CSNK2B than the heterozygous, this green bar shows a big drop off in survival as well. So CSNK2B in the brain specifically has an effect on survival. Getting even more specialized, the EMX1 CRE model targets CSNK2B only in the forebrain, so the front of the brain, the cortex and the hippocampus responsible for learning, memory, higher order functions. Even at that level, we see a difference in survival. It's most pronounced in the homozygous, which is the green line here, showing that you need both, the mice need both copies of CSNK2B in their brain in order to survive. And in the heterozygous mice, you also see a difference in this red line across time. Not only is survival affected, but growth is as well. Here we have the heterozygous mice on the blue bars, and you can see that over time, this is measuring mice by weight, the weight of the mice and their growth is impaired. That's with CSNK2B targeted in the whole body. When we target CSNK2B in the brain using the ICV-CRE model, we also see a similar pattern where when the mice lose CSNK2B, their growth is very severely affected. This matches up with some of what we've seen in the patients as well, with reduced growth, reduced body size, reduced growth velocity as well. In mice, we can also look at different types of motor development and motor milestones. One test here is surface writing. It's an early in life marker where we test whether mice can flip onto their bellies, from their backs. We see a delay in the red bar here, which represents the heterozygous mice throughout the body. There are other measures as well, looking at motor coordination as the mice grow and develop. And we also see a difference in the red line here. You can see it's slightly delayed offset from the blue, showing that CSNK2B in whole body is responsible in mice for motor development. There's also differences in activity that we can see in these mice. I'll draw your attention to the green bars. When we target CSNK2B in brain, the mice are more active and they don't seem to respond to cues in their environment over time, whereas normally we would expect mice to habituate or get used to their environment. These mice remain active when they don't have CSNK2B. We also see this when we're directing the CSNK2B targeting to the forebrain, so the cortex and hippocampus. These are areas of the brain that are responsible for integrating our environment and translating it into actions, and we see differences here as illustrated by the red bars. There's other tests that we can do as well. This is a short-term memory test where we look at their mouse exploration in a maze that's shaped like a Y. We find that when we target CSNK2B in the brain, looking here at the green bar, the less CSNK2B that's present, the less they explore in a normal fashion, and the less they remember where they just were, and that's shown in their differences in activity patterns. This is an example trace here. Talking about gait coordination, we use pretty sophisticated tracking tests where we put mice in a program that tracks their movements and their gait. This is analogous to the types of motor tests that are done clinically, looking at whether which muscles patients are able to use properly and whether they're able to coordinate their gait. And you could see in this line here with the green, mice that have less CSNK2B have a very different footprint pattern, showing that they have a very different way that they're coordinating their gait. Continuing on the subject of learning and memory, we have some preliminary evidence here. I'm going to show you this red bar that certain of our heterozygous mice, when we target CSNK2B in the whole brain, are less able to learn from an auditory cue. Again, this is related to learning and memory and the intellectual disability feature that we see in some patients. And then the sensory response. CSNK2B in the brain seems to be related in, seems to be involved in how mice respond to an auditory stimulus. For example, in the wild, for a mouse, it would be something like a cat. If they make a sound, the mouse startles, and that's how they're able to integrate sensory information from their environment and respond appropriately. We see that this is hyper exaggerated in our mice, and that's shown in the red tracing here. So an exaggerated sensory response. We see that too when we target CSNK2B in the cortex and hippocampus, areas that are involved in this response and sensory modulation. And finally, I'm showing it finally here, but this is important, especially to what we see in our patients. The seizure threshold is something that we test using the electroconvulsive threshold. So we introduce electrical stimulation to the mice, and we see how much it takes for them to have a seizure. And in these red bars here, you could see that when we target CSNK2B throughout the body, there is a reduction in the seizure threshold. These mice are more likely to seize. We see the same thing when we target the entire brain in these mice. That's more seen here in the green bars on the right. They have a lower threshold. They're more likely to seize. And we also see that when we target the cortex and hippocampus, in both females and males, we see in the red bars that the threshold is lower, meaning they're more likely to have a seizure at lower stimulus intensities. So overall, that was a lot of slides and a lot of information, but here's the summary. I'm showing here at the top the EMX1 Cree mouse, which is where we target just the cortex and hippocampus. We see that there's a reduction in survival. There is this increased activity, and then increased startle or exaggerated sensory motor response, as well as reduced seizure threshold in these mice. At the level of the brain, when we target it, we see reduced survival, reduced growth. We see this increased activity picture, and we also see differences in gait coordination, as well as reduced seizure threshold. And finally, when we target the whole body, we see the most broad deficits where CSNK2B throughout the body of the mouse, if it's missing, there's reduced survival, reduced growth, as well as delayed motor milestones and reduced seizure threshold. We don't have final numbers yet on behavior, learning, and memory in these mice. But you can see that, you know, having these mouse models, while I'm talking about numbers, they allow us to get to reach a critical mass in terms of numbers of mice and numbers of subjects that we can't achieve when it comes to patients, since there are so few and the variants are variable in their effects. This allows us to get the numbers to really establish the relationships between brain regions, behaviors, and the changes that we're seeing in our patients. So take-home messages here, what we have found in our mouse models. They reflect the loss of CSNK2B expression in patients. So this mirrors what we're finding in patient cells, and that losing both copies of CSNK2B has profound effects on survival and function. Having one copy of CSNK2B that's normal or wild type in the patients may allow patients to compensate for the loss of one copy, and they compensate in different ways, which can lead to some of the variability we're seeing. And then we see that poor growth is seen with body-wide and brain-specific loss of CSNK2B. Along with growth, there are differences in activity, motor coordination, sensory processing, and memory tests. We see these differences clinically, and we can test them in our mouse models as well. And then in our seizure-related tests, targeting CSNK2B lowers the seizure threshold. Survival time is also affected. The reason I'm showing that is because there's possible unobserved seizures. We don't monitor the mice 24/7, but when a mouse passes, it could be because they had a lethal seizure. And we also have EEG recordings that are ongoing, and those are giving us some clues as well as to which brain regions where the loss of CSNK2B can cause hyperactivity or hyperexcitability that are related to seizures. And just one more slide to show how CSNK2B mouse models are tools for translational research. Here in this picture, you could see that these brain regions in mice are also related to the brain regions in humans. And I know that some of these words that we use for mice are familiar to some families as well. The cortex is where EEG leads are placed when we're looking for seizures. The hippocampus is where seizures also affect and can be generated from. And also important for learning and memory. These are areas that we can get to studying in mice in ways we can't do in humans. I'm mentioning the phosphoproteomics project here, just like Dr. Prikova is doing it in human derived cells, we can also do it in mouse tissue, again, generating the numbers that we need to make some conclusions, as well as getting ideas from mice that we can then test in human cells to see if those same principles are holding true. We can also get brain tissue and test what the brain structures look like in these mice that are missing CSNK2B to see whether perhaps these areas look different in cortex and hippocampus that are related to these behaviors and these signs of seizure susceptibility that we're seeing, and also test those ideas in patient-derived tissue. And then, of course, what we're looking for in the end is developing therapies for CSNK2B-related disorders. A gene does lead to changes in a gene causes changes in biological processes, which then cause these changes in clinical features. We can improve these. We could test improving these deficits in mice by delivering the CSNK2B gene, a normal version, by restoring those in mice through injection or introduction in other ways. We can also, by understanding the biological processes that are involved in these, target those processes using drugs and other approaches, and those kind of converge to hopefully help the clinical features and the difficulties that patients have with these disorders.
Thanks, Abigail. That was wonderful. So if you stop sharing, I'll share. I want to say thank you. First, thank you to Ia and to Abby for reporting on their work. Just pull up my slides again quickly. So it takes a lot of people to do the work that was being presented. And I want to thank especially Ian, Abigail, but also Natalie Lippa, who took the lead on the gene reviews work that you saw. We have a lot of newer people in the lab who, a lot of newer people in the lab who are engaged in the work, and also a resident who is going to be looking at the patient data with us. Christopher Mackinson and his laboratory have been critical collaborators in this work, and really it's a team effort between the two labs. And our external collaborator, Daniel Schwartz in his lab, and Audrey Weber over there, are going to be critical to the biochemical characterizations that both of the speakers that you heard from, that's where that work is going to be done. I have to say, thank you so much to the foundation for providing a platform to discuss CSNK2B with families, providing resources to families, and really all of the families for their generosity in getting involved in research. And if there are those of you who are interested in research and who haven't been involved, please contact me, please get in touch. And then, all of the work that you saw done is being supported by a generous gift from Frank and Heather Jackson, without which really this could not have been possible. And so a lot of thanks to pass around. And then I think we should do questions. Thank you.
Thank you so much. I'm going to stop recording really quick, but yeah, thank you so much to everyone that has worked on this. It's really incredible and exciting to see how much has happened in just a short time. And yes, big thank you to Heather Jackson and her family, because this is possible because of them. So we're grateful to all of you for your work. I'm going to stop recording and we can do some questions here. All right, I have stopped.